I'm Rashmi from Neuroscience Hub. Today we are going to interact with Dr. Pankaj Suman, Scientist E from National Institute of Biotechnology. Today he is going to explain us about snake venom.
I am Dr. Pankaj Suman, working as a scientist E at National Institute of Animal Biotechnology. We have a laboratory over here which is focused on doing research in the area of developing point of care diagnostics and novel therapeutic interventions for one of the key aspect in from Indian context, which is the snake envenomation. And if you for audience, so I want to convey that it's biggest problem in India and also across the globe. And what problems what we are facing in the snake bite management is at the primary healthcare center there is no point of care testing facility available, and there is no devices and diagnostics available for early diagnosis of snake bite if that happens. And because of that, a crucial time point when someone can take a decision and quickly rush to the secondary and tertiary healthcare centers, it gets delayed, and because of that many a times people lose their life. So considering that and research gap in the field of snake bite management, our team has developed tools for early detection of snake envenomation at the point of care testing facilities. So in that direction, we have developed antibodies, monoclonal antibodies against different biomarkers, and we are also working towards development of aptamer, which is a small nucleotides. It's not a protein, it's a small nucleotide that can be used for diagnosis of venom if that is present in the biological fluids. So what we are doing is, one side we are developing a point of care diagnostic tool for snake bite management, and also that at the primary healthcare center, or so we can get the rapid diagnosis, a specific rapid diagnosis, so that specific treatment modalities can be followed at the clinics. And second one, we are also working towards development of highly sensitive sensors that can help in identification of amount of venom in the blood circulation when patient is reaching to the hospital. Because for snake bite management, if you see the trend, then 8 to 10 vials, even more than that, of antivenoms is used for management of that. And to rationalize the antivenom therapy, it's very important to know that is it possible that we know that how much venom is circulating without neutralization, and if we know that how much venom is still has to be neutralized, then we can rationalize the antivenom therapy. For that sake, we are working towards developing electrochemical sensors that can tell the clinicians in the real time when they are admitted into the hospital that how much antivenom to be administered, and if that gets rationalized, so we can reduce the side effects which is there because of the antivenom administration. We can reduce that. So overall, our aim is to reduce the suffering of the individuals in those who get problems because of the snake bite. And for that, our lab is working towards developing point of care diagnosis as well as clinical diagnosis through development of ultra sensitive electrochemical sensings. Another research focus of our lab is to develop a new generation of antivenoms, where we are planning to use aptamer based technologies for development of antivenoms against major snake population in the India, like all the big four snakes specifically in the beginning, and then we will move forward for developing the technology for other toxins or other venoms. In our lab, we are making different kind of devices for like different kind of problems which we are trying to address and making the point of care diagnostics. And we are using 3D printing technologies for addressing you can make different kind of devices as per the requirement. for the different kind of assays to be portabilized, and that can be used at the point of care testing facilities. For that purpose, what we are doing is, one of the device which we have made with using 3D printing technologies is for detection of somatic cells in raw milk, and for that we have developed a technologies that has the quantitative somatic cell measurement capabilities, which is equivalent to flow cytometry based assays, and is as robust and as field applicable as a California mastitis test, which is used for qualitative assessment of somatic cells in the field. Similarly, we have also developed a combined colorimetric and fluorimetric device that can be used for rapid antimicrobial sensitivity testing under the field condition. So why these two tests are very important is, if you use the somatic cell counting, that can help in identifying the animals who is having a mastitis at subclinical or clinical level, and if you identify them, then you get a chance to select the appropriate antibiotic within half an hour to 1 to 2 hours time, and give rational antimicrobial therapy to these animals, so that you can reduce the burden of antimicrobial resistance that gets transmitted from animals to human being. So for that sake, the 3D printing technologies and the instruments which we have is helping us in a great way to develop a portable devices and the kind of design which we want to use at the field level. This work was featured by Nature India. Okay. On the editor also did a podcast for this device, apart from highlighting that on Nature India website. And after that, Hindu wrote half-page write-up on this. We will give you a write-up for this technology which we have developed, all this. Then is it possible to make a for educating people that how somatic cell count at home is so important, and if you use such kind of technologies developed, then that will help you, that when you are using that good quality milk at home, then your health and well-being for the family.
Myself, Bratati Rai. I'm from VIT Vellore and working as a dissertation trainee here. Here I'm trying to develop an electrochemical sensor which would help to detect venom in blood serum samples. So these are the sensors. Here we would be adding the blood serum samples, and it will have the capability of detecting the amount of venom within 10 minutes. If the venom level is detected, then the line here, if you can see these graphical lines, they will be going upwards. So within 10 minutes, the venom can be detected in blood serum samples of any patients.
Thank you very much for listening, and looking forward to share more information of snake bite management at the point of care testing, and for the farmer oriented technologies which will come out from the lab in future.